Hello students, in this part we will discuss on RNA world and the replication process. RNA world. What is RNA world? RNA was the first genetic material because there are many evidences are present which show the essential part of RNA in our life processes such like as metabolic process, translation, splicing. So first of all, what is the RNA world? RNA was the first genetic material. This is because there are many evidences present which show that many essential life processes are depend on RNA, such like as metabolism, translation, and splicing. These are the processes which depend on the RNA. Second one, RNA is an adapter structure molecule which is also a catalyst. RNA is an adapter structural molecule which act as a catalyst. Being a catalyst, RNA is more reactive. Being a catalyst, RNA is more reactive, and we know that the substance which is more reactive that cannot be remain stable for a long time. So it does not stable, or we can say that it doesn't show the property of stability. In place of RNA, if we comparison RNA with DNA, in last section we have already discussed, we have already compared RNA with DNA. But if we comparison RNA with the DNA is double-stranded particles and which has stable chemical components not present in RNA. So DNA is known as genetic material. This is the property which show that there is no stability present in RNA. So we cannot calculate the RNA is the genetic material. But DNA, which is double-stranded, as well as DNA contains stable chemical components which are not present in RNA. So when we calculate, when we examine the genetic material, we prefer DNA, not RNA. This is the RNA world. Now next part, what is replication? Replication means formation its copy. DNA form its copy. formation DNA complete copy, that is known as replication. The next one is replication. What is replication? Replication is formation of its copy or formation of copy with the RNA or DNA genetic material. DNA it makes its copy, that is also known as replication. RNA makes its copy, that is also known as replication. Replication we will discuss on replication of DNA. DNA shows two types of processes. One is autocatalysis and second one is heterocatalysis. DNA represent two processes, such as one is autocatalysis and second one is heterocatalysis. Autocatalysis, it means DNA make its complete copy and that forms DNA. DNA produce its complete copy, DNA change to DNA. We know that DNA is a double-stranded particle. When we separate, unwind both the strand has the ability to maintain its copy and to form its copy by the DNA, that is known as autocatalysis. This process is also known as replication. In this part, we will discuss on this process. And second one is heterocatalysis. Heterocatalysis means DNA produce RNA, DNA change into RNA. And the process in which DNA change into RNA, that is known as transcription. In this process, DNA change into RNA. In this, DNA to DNA, same. So it is known as autocatalysis. Hetero means other, DNA change into RNA. This process is also known as transcription. This process we will discuss later part. Firstly, we will discuss on this part, replication. Before replication, there are some enzymes are required. These enzymes help in the replication processes. Replication process, what are those enzymes? Those enzymes are the following. First one is helicase enzyme. What is the role of helicase enzyme? We know that DNA is double-stranded particles. Both the strands are twisted, coiling over each other. What is the role of helicase enzyme? This helicase enzyme unwind these both the strands. Unwind means to break down the hydrogen bond between the two strands and unwind it. So what is the role? First of all, helicase enzyme. This enzyme unwind the double-stranded, or you can say double strand of DNA by breaking the hydrogen bond. The second enzyme is topoisomerase. Topoisomerase enzyme. As we have already discussed, DNA is double strand, both the strands are twisted over in each other with tight force. But with the help of a helicase enzyme, when we unwind it, there are some tightly force also attract with each other, so they form the tension. Whenever we take the two ropes and twist it with each other, wrapped in each other tight force, we unwind it. You can see that there are some folded may be can change, some folded may be produced. This tension force may be stopped by the topoisomerase enzyme. It means to provide tension, or you can say that to protect the tension force. It means to separate, to remain separate both the strands from each other. To remain both strands separate. So topoisomerase enzyme is required. After that, as we use topoisomerase enzyme, both strands have some special protein so that they maintain their stability, no change in the single strand. Next one is single strand stability or stable protein. This enzyme, first helicase enzyme unwind both the strands. Second topoisomerase enzyme, it remove the tension force so that the both strands remains separately. And when both strands remain stable, separate and they remain stable, there is some other protein required. This protein maintain both the strands stable. Next one is ligase enzyme. DNA makes its copy, but not continuously. There are some gaps present. There are some cuts present between the DNA particles. These fragments of DNA are adjusted, are joined with the help of ligase enzyme. This enzyme, this helps in joining the fragment of DNA. Next enzyme is DNA polymerase 1. This is the first, second, third, fourth, fifth. Fifth one is DNA polymerase 1 enzyme. What is the role of DNA polymerase 1? We know that whenever we separate, unwind both the strand of DNA, both strands have the ability to maintain its copy, or we can say that both the strands have the ability to make its complementary copy. But this complementary copy may be started after the adjustment of primer. So that primer is required after the formation of complete copy removed. To remove the primer, we need DNA polymerase 1. So what is DNA polymerase 1 enzyme? This enzyme remove the primer. What is primer? The primer is synthesized by the primerase enzyme. Next enzyme is primerase enzyme. This enzyme is responsible for synthesis of primer. Whenever we adjust primer at the strand, start to make its copy. First of all, primerase enzyme is required rather than the DNA polymerase 1. Because required synthesis of primer, attachment of primer, then the complete copy of DNA can take place. As the copy of DNA completed, DNA polymerase 1 is adjusted so that it removes the primer. Primerase enzyme to synthesize primer. Second one, it is the segment of RNA. Primer is segments of RNA, but it dependent on DNA. To synthesize primer, this enzyme can take place. What is primer? Primer is a segments of RNA. So we can say that primer is a DNA dependent RNA particles. DNA dependent RNA particle, that is primer. Next enzyme is DNA polymerase 3. Polymerase 3. All these enzymes except then DNA polymerase 3. These enzymes help in synthesis or help in the replication. But this is the only enzyme which is responsible for synthesizing a DNA copy. This take in the replication, but they help only. DNA polymerase 3 enzyme, it contains 3 components. Alpha unit. This alpha unit act as a catalytic property. Second one is epsilon. This epsilon unit is used in proofreading. Means how the DNA makes its copy or not. Proofreading. And next one is beta unit. This beta unit is responsible for sliding clamp. It means formation of DNA one strand copy to another in the sequence. Sliding clamp. Next enzyme 8, that is phosphorylase enzyme. This enzyme is responsible for adding the phosphorus as the requirement of the chemical substance to adjust or to add phosphorus as required by the chemical compound. And last enzyme, that is nucleases. Nucleases enzyme is also act as proofreading. So we can say that there are 2 enzymes which act as proofreading. One is epsilon, which is a part of DNA polymerase 3, and second one is nucleases enzyme. This enzyme is also responsible for proofreading. So before going with in the replication process, first of all we need to require how these enzymes are required, what are the function of these enzymes. The enzymes which are required before replication, those are, first one is just like as helicase enzyme which unwind both the strand of DNA, topoisomerase enzyme after removal of the hydrogen bond. There is some tension force also present. To remove this tension force, the topoisomerase enzyme is required. Single strand stable, there is some proteins which are act as enzyme. That protein helps the single strand of DNA as they separated to maintain their stability. Ligase enzyme, as DNA start to make its copy, it is not continuously, it is fragment. So ligase enzyme have to adjust to add the fragment of the DNA. Next one is DNA polymerase 1. We can say that this enzyme remove the primer, first one, and filling the gap. Because as we remove the primer, there is gap present. That gap is filled by the DNA polymerase 1. Primer is a RNA segments, but it depends on DNA. So we can say that primer is a DNA dependent RNA molecule. Primer is synthesized by the primerase enzyme. Next enzyme is DNA polymerase 3. As we discussed, there are 9 enzymes required in replication. Out of 9, one enzyme which act as synthesizing of DNA particle, that is DNA polymerase 3. All rest 8 enzymes only help. This DNA polymerase 3, it contains 3 units: alpha unit, epsilon, and beta unit. Alpha unit act as catalyst, epsilon is a proofreading, and beta, it helps in sliding clamp. Phosphorylase enzyme, if there is a requirement of in the chemical compound phosphorus, this phosphorus is added by the phosphorylase enzyme. And next one is nuclease enzyme. This enzyme is also responsible for proofreading. In next part we will discuss on the replication process.